Welcome everyone. It's 9 a.m. PST, 4 p.m. GMT, and 12 p.m. EST. So I'm delighted to be opening another Global IMMUNOTALK. First, I would like to remind you that our next speaker is our co-organizer that is with us today, Matteo Iannacone, and that will be next week. And so I hope that you can join. And also, I'm delighted that today is with us our global immuno speaker, Dr. Andrés Hidalgo, that will be now introduced by Matteo.
Thanks, Alina. And on behalf of all the Global IMMUNOTALK organizers, it is truly a great pleasure to introduce Andrés Hidalgo as today's Global IMMUNOTALK speaker. Andrés is originally from Madrid, Spain, where he obtained both his Bachelor of Science degree in biology, as well as his PhD in hepatology from the Universidad Autónoma de Madrid. He then moved to New York City for his postdoctoral training and joined the lab of Paul Frenet at Mount Sinai. As a postdoctoral trainee, Andrés developed and used live imaging modalities to study acute inflammatory diseases and discovered the receptors that mediate early neutrophil recruitment, as well as the signals that cause vascular occlusion in sickle cell disease. In light of his accomplishments, Andrés was promoted to assistant professor and remained at Mount Sinai for an additional 3 years, prior to being recruited as a team leader at the Spanish Cardiovascular Research Center in Madrid. In his lab, Andrés further developed live imaging technologies and applied them to the study of thromboinflammatory disease in several organs, including the lung and the heart. He also discovered new functions for innate immune cells and demonstrated that circadian rhythms in the bone marrow are entrained in part by neutrophils entering this organ. He identified immune circadian clockworks that control leukocyte migration, transcription, and release of toxic mediators, and underlies circadian defense and inflammation. More recently, he has extended his analysis to tissue-resident macrophages in multiple organs, including the heart, and used imaging to identify mechanisms of mitochondrial homeostasis. I find Andrés to be a very creative scientist that combines cutting-edge imaging technology to answer interesting biological questions. And for his contribution to science, Andrés has received numerous awards, including a Development Award from the American Heart Association and David B. Paul Prize for Innovative Research in Transfusion Medicine. He has been a fellow of the Ramón y Cajal program, and he sits on the editorial board of Blood. And Andrés has given me permission to announce that he has been recruited to Yale, where he will soon join the Department of Immunobiology. So many congratulations and good luck with your move. Andrés, we are very grateful that you have accepted our invitation to speak today.
Thank you guys for the invitation. Thank you for the moving this initiative forward. I think it's a relief and it's a breath of fresh air in these pandemics to continue exchanging ideas and knowledge on immunology. So thanks very much, and I'm very happy to see good friends also on the other side.
Our pleasure. So Andrés, before you start your talk, it's our tradition to ask speakers a question to get to know them a little better. So the question we would like to ask you is, which scientist has inspired you the most in your career and why?
That's a tough question. Although I knew a bit about the question already, I gave it some thought because I thought it might be an interesting thing for younger people to know. So I don't have one. Like everything is in immunology is very granular now. I have several. I thinking about this, I realized that the choice of my immunology or scientist heroes are those that fulfill the 3 criteria that I think a good scientist would have. The criteria are focus on an important question, creativity and innovation, and mentorship. So when I think about a drive for a question and being able to push and pursue new ideas, I'll give just names, but of course there are many depending on your interest. So my references have been Porphynet and a younger one, McCann Gilliams, whose love and his work on niche and how the environment specifies the cell type, I find just fascinating. So I think in many ways this is shaping the way we do immunology now. In terms of creativity and technological advance, I have my favorites. I think single cell transcriptomics is just come a lot to our work now, or single cell biology, not only transcriptomics, and I think the work of Ido Amit has been very inspiring for me. In terms of creativity, I love, and coming from imaging, I love the work of Michael Sext. I just find it an extremely creative and elegant scientist. And for mentorship, each one of you in each field, probably you have your own favorite, but of course I always think about people like Erf Weisman, Miriam Merat, people who have put, or even Uli Bonandrian, people who have seeded a lot of great scientists around. I think that gives credit also to their work in a different way. So yeah, it's not one, but I think I mentioned 7. I have many, and I think that would pretty much summarize.
Thanks, Andres. This was very nice. So we are now very eager to listen to your presentation. So if you want to share your screen.
Absolutely. Yeah, can you see now?
Yes,
Perfect.
you may want to, yeah, you can see the small slides on the side.
Yeah, okay, so. All right, it's all okay now? Can you see the presentation mode? All right, so thanks again, Elena, Mateo, Florian, Enrique, Carla, everyone involved in this for the invitation. It's an honor because I admire, as I mentioned, many of the speakers that you have invited. So it's just humbling, and I hope that I'm up to the level. And mostly the idea is to give a little bit a vision of the type of work that we do, the way we do science in the lab, and also inspire a bit of love towards one of our favorite cells, the neutrophils. There was recently this contest in Twitter that I found quite interesting and fun on what is your favorite cell type, and of course there was some debate, some fun debate there, and it was great and enlightening because you could see how much weight and how people envision the different leukocyte subsets, and I have to say the neutrophils didn't make it very far. That made me a little bit upset, but I appreciate that we need to kick up a little bit on the appreciation of this. So I hope our work inspires some of you towards this. So I want to talk about the power of N was the original title. The neutrophil is disguised. Basically, it's the same idea. Is how we are starting to see a different biology for neutrophils beyond the classical view. Same thing that happened perhaps 20 years ago with macrophages. So it turns out that there's a lot of similar ideas with neutrophils, but with some peculiarities, because clearly the cell cycle is quite different. So that gives some nuance on how these cells contribute to many aspects of physiology and how they disguise, adopt different identities, if you will, or states to accomplish this. So just start with a view on general view ideas of immunology now compared to when I started doing immunology. So many things have changed, and one perhaps of the most prominent ones summarized in this image from McCann's review is that the strict hierarchical division of the hematopoietic tree, which could apply to any cell lineage really, it is changing a lot. And what we see is that rather than a strict and predetermined divisions, there's hesitations, there are transition states between cell types. So you cannot draw really straight lines coming up from above, but there's a lot of decisions, some of which are influenced by the environment or by the state of the organism. So really it creates a lot of versatility and plasticity of the system, which is a key, I think is the key word in how we see immunology and biology in general. There's much more plasticity the more we understand the granularity of the system, the more we appreciate this. So this is an example of this, and perhaps the living cell type in these efforts, at least in the myeloid lineage, have been macrophages, and we do some work on this in the lab, but in general, it's been appreciated for over 10 years now that there's a lot to be said about our original view of these myeloid cells. So they're really under a single name. We encompass a number of different macrophage cells with different functions, even if they might share the same lineage or not, depending if they come from an embryonic or postnatal origin, and really how the tissue influences the biology of the cells to accomplish different tasks. So they're immune cells, but really they're not doing immune functions. And these concepts are very much influencing the way we do our work on neutrophils and asking whether something similar applies to this other cell type. So to illustrate this idea of fates and identities, I like to put this slide with two of my immunology friends and heroes, Florent and Martin, sort of to illustrate how the perception of the individual is important. So as humans, we can discriminate these two guys quite well because of the beard, the shape of the eyes, everything that you are accustomed to recognizing in another person. But for let's say an extraterrestrial, this would be very different because they're not used to identifying the right traits in a face. So this is a little bit the same problem that we have as immunologists. We don't really know what the real phase of a cell is, if it's the identity of the cell, which would be, let's say, the Florent identity and the Martin identity, or is it the environment that conditions different flavors to each one? So for example, in different tissues, they would adopt different granularity, if you will. So you really need to be trained to recognize that the identity is more important here or here. But perhaps for a person from outside of the Earth, it would be more important to see this type of shade here or this type of glossy shade here when they define the properties of a cell. So this is the same sort of issue that we face when we describe ourselves. Of course, we need to push forward to publish, and in some cases, we don't pay enough attention to this problem. But this really is a key question, I think, when we try to move forward the biology in this decade. A few years ago, together with Renato Stuni and with Lai-Wan Ang, we defined, following the same ideas of what defines a cell subset, not different subsets or different identities, but really cell states or subsets of neutrophils, if there's any. And we came up with a number of criteria that could be used. Of course, this can change, but we thought that the epigenetic and transcriptional profile was important, but also things that were not so clear not too long ago, like where are they located, the phenotypic profile, maturation status, site of origin, effector function, and so on and so forth. So really not an easy question, and I leave it just out in the air, and I think it's something that is important to think about because it will condition how we plan our experiments and how we move forward. What is it that is more important when we think about our favorite cell type in disease, for example? So I'll talk one of my favorite topics, which is following on this idea that myeloid cells are heterogeneous and adaptable. An example of adaptation comes from the field of circadian oscillations, circadian biology, that has a long tradition and has focused a lot on the molecular basis for this, which is really fascinating. And there's not been, until relatively recently, so much work done on how circadian oscillations, so changes in the environment, influence myeloid cell biology or immune physiology in general. So just to give you an idea of why this is important, what we're seeing here is adaptation to an environment. And in our case, we live in this planet, in this ball that happens to turn around the sun, which is the source of energy and light. And as the Earth turns around, it creates environments that change. They change in temperature. Our predators wake up our food becomes available. And so our organism in many ways needs to adapt to this. So it takes as much profit at this as possible. But this creates, of course, challenges because the physiology on one side and the other of the planet is not the same. And so immunology and many aspects of physiology, for example, metabolism, cell replication, and so on, change to recognize these challenges as they come. So what about immunology? And in a recent review, we gave a thought about this, and what we thought it would be more interesting. What we realized on reviewing the literature is that rather than looking at specific changes on an individual cell type, depending on the night dark light cycle, was that these really appear to be very much integrated into the physiology of the different organs. So we know that, for example, light dark signals imprint on the central nervous system, and through nervous signals or humoral cues, this transmits these night day cycles to the other cells, sort of to synchronize the system. But what we realize is that there were very few and rare examples where you could isolate an immune cell as being the target of these circadian oscillations, but rather whole systems appear that communicated with each other to establish what we call circadian immune circuits. So for example, signals that originated from light or from feeding that imprinted on what we call the structural cells and epithelial cell or mesenchymal cells, fibroblast, that produce factors that in turn regulated the function of an immune cell, which in turn exerted a function on a tissue or organ, either in inflammation or under steady state conditions. And there are good examples of this in the literature. I won't go into details, but basically many, if not every aspect of physiology of your favorite organ will be in one way or another influenced by one of these circuits. So this is something also to keep in mind when you do your experiments and you try to stick to a specific time of the day, but also when you think about how you interpret your data. So in thinking about the circadian influence on the biology of neutrophils, one simple experiment that we did a while ago, and Maria Casanova was leading this work, is that we realized that simply looking at the phenotype using only two markers, you know, back on the days when you use only two markers to do an experiment, that we could classify two different types of cells depending on the levels of CXCR4. L-selectin, we define this L-selectin low population as an H neutrophil, and the reason we call it H is simply because more time had passed to generate this population. So the ones that come from the bone marrow are the gray cells, and then they transition into the red phenotype. So that's the reason we decided to give this name. But anyway, you could see that there were changes that occurred during the different hours of the day, right? This is taken every 4 hours. We take a sample and we analyze it. Only by doing this and not influencing in any other way, we already saw changes. Now the question is, what control these changes in the phenotype of the cells? So the phenotype, as you see, also conditions many aspects of the function. And it was quite a few years later that when Jose Maria Drover and Maria initiated the studies came that we could sort of provide a solution to these intriguing oscillations. And what we discovered is that a lot of these changes are intrinsically encoded by the leukocyte. Of course, with influence from the outside, but depending on genes that are only expressed on the neutrophil. And what we realized by doing transcriptomics or chip analysis is that there was this basic mechanism underlying this process, whereby this BMAL transcription factor, which is a core circadian core transcription factor, regulates the expression of many genes. But one of the genes that it regulated in neutrophils was this chemokine CXCL2. And what we found is that through a cell intrinsic or cell autonomous mechanism, stimulated the cell through this receptor CXCR2, and these drove, you know, this, if you will, low grade activation of the cell, induced this process of aging, the circadian activation, if you will. That's I think a good way to define what aging is. But at the same time, in additional experiments, what we found is that there was also a negative hand to this clock, which is CXCR4, which negatively interacts with this pathway, possibly by contracting the activation of CXCR2. And this is interesting because now we have, as you will see later, this creates some opportunities. We have a positive handle that runs the clock, the circadian clock, and one negative regulator of this. So one, I think, good example of how this influences the physiology of the cells is by creating animals in which we specifically targeted either CXCR4, the negative regulator, or BMAL1, the positive regulator of this, in a neutrophil-specific fashion. So all the cells of the animal are wild type. It's a neutrophils which like one or the other element of this clock. And when you do this, what we found is like compared with the typical oscillation that you find in wild type animals, this is the aging phenotype that changes during the day. If you eliminate BMAL1, if you interact here, then the cells cannot engage into this phenotypic change and they remain flat throughout the day. And if you eliminate CXCR4, then they really cannot block this process and the cells are permanently aged in this phenotype. So that's how we think about this process to be deterministic and regulated specifically by this molecular clock. Now, there are many examples of how this influences physiology, and I'll just show a few of them. One that I thought was quite interesting was that the shape of the cell, the aspect of the cell, really changes quite dramatically. So this is what we call the night neutrophil, right? When we would be here in this phase in the dark cycle, and the day neutrophils. We haven't done anything to these animals. We just take the cells and look at them by scanning electron microscopy. And what Jose found is that these cells were much more hairy. They have many more of these microvilli than day neutrophils. This, of course, already tells you that the cells are different, but coming from the field of live imaging and inflammation, a quick idea was that because a lot of the receptors needed for recruitment into inflammatory sites are accumulated on the tip of these microvilli, this in fact could mean that these cells are much more sticky and prone to go to inflammatory sites than these other cells. And this is the experiment that we did to demonstrate this, where we created in the same animal, we put together cells, we used a trick, I won't go into details here for the sake of time, with cells that have this nighttime phenotype, the green cells, and cells that have a daytime phenotype, the red cells. And they are at the same, more or less the same levels in the circulation. However, see how many more of these green cells roll on this vasculature. And this is because they have more of these microvilli. So if they do more rolling, then you expect to see more adhesion, which is exactly what we see in the vessel a few hours later. And if you wait long enough, there's not only more adhesion, but also more extravasation. So in the end, this is what you care about, how many cells are able to go to the site of inflammation. So this, think in very simplistic terms, this differential recruitment is regulated by the control of these circadian changes and inside the neutrophil. So this is a cell intrinsic phenotype, as also some examples later. So this is this is more or less the model. In the steady state condition, both cells, and this is, I won't go into details into this either, in a vessel that is not inflamed, there's recruitment of both cells, even if there's no inflammation, but this is a different story now. Now, if you have inflammation, then the night-like neutrophils are much more prone to do this rolling and to accumulate into the tissue rather than the daytime neutrophils, which don't have this microvilli. And this has, of course, implication of how the immune responses will be between night and day. So this is an example of this. What we did in the lab is to infect a wild-type animal with Candida albicans. It's something that has been done before, and then look the differential response if you did the infection at daytime or at nighttime. And as you can see here, the protection at nighttime is better. We're measuring here weight loss, so the more weight that is lost, the more severe that infection is. And you can see here that if you do an infection at daytime, then the animals get sicker. Now, what happens if we eliminate, disable specifically this clock in neutrophils? And what happens is that this sensing of time is gone, basically. The response is exactly the same. It's not that we go into an immune deficiency or exaggerated inflammation. It's simply that the small difference in response is gone. And one could argue, what is the relevance of this? Again, I'll show you some examples of why this is important. But the idea here is that you don't create really strong susceptibilities to infection or inflammation. The system is still working. It's just that a small increase in activity might be beneficial if you think in wild animals or in humans many thousands of years ago. to defend against subclinical infections, to contain things before they happen. So that's a little bit the interpretation of this rather striking finding. So a little bit to summarize this series of studies, the idea is that this transition, this heterogeneity based on time of neutrophils between night and day, which we refer to as aging, might be important, and we argue that it's important to take maximal advantage of the specific differences between the cell types. So the nighttime neutrophils in the mouse, at least, they're better to fight, to go to sites of inflammation, whereas the daytime neutrophils that have lost the microvilli, they are perfectly fine to go to non-inflamed tissues, but they have trouble to go to inflamed tissues. And of course, the other changes on these cells that overall, we think, is that gives an advantage to having these cells circulate during nighttime and these cells circulate during daytime. And it creates this sort of oscillation. So now with these genetic mutants that we generated, we can sort of appreciate, even if we cannot run the experiment in the wild, which is what I would have liked to do, but that became quite challenging. So if we eliminate one gene or the other, we go into the extreme. So either a constitutive fresh state, so the animal is somehow protected from self-injury during sterile inflammation, but there's also a loss of the elevated response against potential pathogens that invade. So that's one trade-off that you see here, and the opposite is here. Here you have a good defense against pathogens that might invade the tissues, but at the same time you're in a elevated state of sterile inflammation, and this to compensate for this, or to be able to take the best of both worlds, is what we think the circadian cycles are useful to benefit from the specific times when one or the other function are most needed. So that was a lot of the work that was based on looking at the transcriptional profile of neutrophils. And one particularity of the cells is that rather than, when we're thinking about acute responses, rather than initiating transcription, which they don't do very well, as I will discuss later, they move proteins, because one thing that they do during the maturation in the bone marrow is to accumulate a lot of the proteins that they will need for antimicrobial defense in granules, so they're ready to go. The cell gets activated, the granulates, and there you go. You have your immune response that does not necessarily depend on transcription. So this has advantages for the cells that are frontline defense, right? So they have the weapons ready to go. So we thought that it would be important to understand not only transcriptional responses, which, you know, identify the pathway that I just discussed with you, but also to look responses at the protein level. And this is what Jose Maria Adrover also took the challenge of. So the idea here is that what we did is to compare the proteome rather than the transcriptome, the proteome of many cells that we purified over several months, to compare what was the differential proteome between nighttime and daytime neutrophils. And what we found, quite surprisingly, not in retrospective, but at the time, was that there were many more proteins that were enriched in the nighttime neutrophils. There were more protein enriched when you compare them to daytime neutrophils, which would be this volcano plot here. And these proteins really are part of many of the compartments of the cell. But in particular, we thought it was interesting because, as I mentioned before, a lot of the antimicrobial proteins components are stored in granules. We thought it was interesting that this applied also to granule proteins. So proteins that are stored in granules are more abundant at nighttime than at daytime. And in practical terms, this meant, and this was surprising to us, that these cells were degranulating spontaneously. So remember here, like before, we do not do anything to the animal. We don't induce any sort of inflammation or infection. We just take the cells at different times of the day. And without doing anything, there's already degranulation, which is a little bit against the dogma that you need activation for the granulation of the cells. So anyway, not only this suggested that the toxic content of the cells was reduced at daytime, so the cells might be less immune competent in a way, but these are also implications for those in the field that are interested in these very interesting curious structures called nets, Probably most of you are familiar with these neutrophil cellular traps, which are released into the environment, and they're great for trapping microbes, but they're also quite toxic because they have a lot of nasty proteins that can be toxic to host cells. So what we thought is, if there's a difference in granule content, there's probably also a difference in the capacity to form in nets. And this is what, again, Jose Maria did, in this experiment. He induced injury into the lungs of animals, acute injury, and then visualized neutrophils in blue, and the formation of nets, which are these red pieces stained with iodine, proteome iodide injected into the animal. So he can visualize the production in vivo of nets or net-like structures. So this is at daytime. This is the number of nets, low rate, even if you induce inflammation. If you do the same experiment at nighttime, what you get is not many more neutrophils. What you get is neutrophils that are much more active at producing nets. And you can quantify this, which is what is shown here, the production of nets after induction of injury. These are nets because we can block this with chloramidine. And in practical terms, this is a model of acute lung injury. What is the consequence towards the health of the lungs of these animals? And the consequence is actually important. If you induce the challenge at daytime, this is nighttime, and here we're looking in red accumulation of water in the lungs, or edema. The response is quite significantly higher if you do the experiment at nighttime than at daytime. And again, this is chloramidine sensitive, so it's net dependent, and this translates into differential survival. So there's an important difference. Of course, this is in the whole animal. We don't know if neutrophils are the only ones mediating this response, but now because we had this molecular clock sort of figure out, we use this genetic trick to use the BMAL mutants and the CXCR4 mutants to show that the same differential response can be seen if you use these mutants. BMAL, which are night-like neutrophils, have an exaggerated production of nets compared to daytime neutrophils, which are the CXCR4 mutants, and this translates into a very marked difference in the formation of edema. The BMAL mutants have much more, whereas the CXCR4 are largely protected, and the survival reflects this difference. So really, the production of nets in the granule content is differentially, it changes over time in neutrophils and follows circadian pattern, and this has implications on when a disease is more severe. Now, this is interesting, of course, thinking in humans. We know that inflammatory disease is circadian. Many of our physiology and disease as well is circadian. So we plan a small experiment to do the same measurements, taking only blood from a number of volunteers, and then looking at the same parameters that we were looking in the mouse. Bottom line, it's the same increased content of proteins and granule proteins was elevated in the early morning, in this case, compared with the afternoon in humans. And even if humans, there's a lot of variability between us because the genetic background and behaviors are so different, we still saw the same response. A change in the granule content, a change in the production of nets, and this is what is quantified here. And perhaps a way to put this, and using again the first icon of these guys, is that here the neutrophil we discussed is really acting as an antimicrobial, a pesticide sort of agent that is trying to get rid of these pathogens. And with this work, what we found is that this disarming, this loss of incredible loss of toxic components creates a level of heterogeneity, so that night and day neutrophils increase responses, neutrophil function changes at different times of the day. So one idea that comes from this, and we're thinking about this process of aging quite a bit, is whether this process and these physiological oscillations in the activity of neutrophils, and you saw that example with acute lung injury, can be harnessed in any way to protect individuals. And the obvious idea, and thinking that quite often when we see some association between aging, the aged neutrophils, and inflammatory disease. So one idea is, why not block this process using the endogenous drivers? So rather than just eliminating neutrophils, which of course we cannot do, or using the strong immunosuppressants, why don't we use the natural way of the cell to blunt the responses of neutrophils, so we don't go into an immune deficient state? So the idea or the proof of concept that this might work comes from this series of experiments where we use as a readout myocardial infarction. So it's a model of acute injury emanating from the vessels, but you can easily read in the myocardium the degree of cell death, so you can score how strong is the immune response. So if we use the mutants in which, the BMAL mutant in which we impair this aging process, or we use another interesting mutant, which is the CXCR4 having the WIM mutation. So this mutation prevents internalization of the receptor. So these patients or animals with these cells with this variant, this variant, have an exaggerated response to CXCR4. So it doesn't quite desensitize, and you can basically mimic the same thing. So either you block BMAL1 or you potentiate this pathway all the way, always with the same result of blocking interfere with agents. So if you do use these two different systems, what we find is a protection against myocardial death, so infarct size. And not only this, but there's also a loss of the circadian pattern compared with the wild type, where you have a strong day-night difference. This reduction in BMAL also goes with the loss of circadian variations, and with the CXCR4 WIM mutants, the same thing. This is a blunting, strong blunting of this circadian variation, suggesting that this might be a good way to target and to modulate the activity of neutrophils against acute inflammatory disease. So what Alejandra Roca is student in the lab is doing now is together in collaboration with the group of Gerd Nicolaes in the Netherlands, is synthesize peptides that are potential agonists for CXCR4. These are 3 different peptides that she synthesized together with a commercial peptide that I will show you for ATI. That we can use to treat animals. So this is this pathway is activated in normal conditions, so we don't think that this will create any susceptibility, but that's what we're testing. So use agonists towards this receptor to block, naturally blunt this effect. And before we go into the consequences and infraction, one thing that we wanted to do with this agonist to make sure that really we are targeting this aging process. So this open again the idea that we needed to look into this aging process in a more granular, in a more accurate way. So rather than using a few markers, 2 markers as we did in the beginning, what we're starting to do is use single cell transcriptomics to look for evidence that treatment with this agonist for CXCR4, either the WIM mutant or this commercial agonist ATI or this P3 peptide, that we really introduce a switch into the state of the neutrophil. So as you can see in this transcriptomic analysis, neutrophils are not the most exciting cell when you do this analysis, but it's still okay. Well, the answer here is that there's a single stage, so there's a single transcriptional space, not specific clusters, but we can shift, and I think you will appreciate here with the colors, we can shift slightly the phenotype of the cells, transcriptional profile of the cells when we treat with the compounds in the same direction when we give the when we look at the mutant and when we treat with the agonist. So we think that this provides additional evidence that that we induce a response that is consistent when we either use genetic tools or pharmacological tools. So what is the consequence of treating with these agonists? It is the same thing I showed before, just to show that there's really a readable transcriptional profile when we engage neutrophils with the CXCR4 agonist. But the consequence in the end, which is what we care about in the model of myocardial infarction, is a strong protection, both when we give the commercial agonist or our P3 peptide. In both cases, we engage into this strong protection. So we think we're still exploring this. The idea is that we can harness this physiological circadian-based process, mild differences, to obtain a benefit, a clinical benefit. So following on this idea of heterogeneity in blood, which I introduced here with the aging and with the using of this CXCR4 agonist, you can appreciate here when you look at individual cells that there's a single space for neutrophils, but there really is quite a wide spread of the cells, not in clusters, so the cells still remain the same, at least with the power of our single cell transcriptomics. So the cells are transcriptional poor, it's difficult to get a lot of reads, but still, even with this information, there's quite a bit of heterogeneity that you can see by studying different treatments for mutants. And this heterogeneity came to my mind that one can translate into what we see in blood vessels when we look at neutrophils. And this is from the studies that I did when I was with Porphynet. So, you know, everything links to everything. That when you look at an inflammatory site, this is a vessel treated with TNF-alpha, and we see neutrophils, we see many of them, and there's some sort of coordinated behavior, there's rolling, there's adhesion, but when you look at individual cells, there really is quite a heterogeneous behavior. We got many evidences for this, in this case, looking for platelet binding in red, polarization, where the cells move more, they move against the current, they're rolling, they're already attaching, they're transmigrating. So there really is, in the same environment, a heterogeneous response. So we thought we wanted to harness this heterogeneity again to gain more information, because we thought that in each cell that is behaving differently, there's hidden information that we're missing out. When we think about transcripts, it becomes more easy. You express more of a gene, then I can tell something about that particular cell. But here, we don't have that information, but we can tell that this cell is moving faster than this other cell, or is going for the flow, or is bigger, or is smaller. So this is all biological information that is typically lost in imaging experiments, and we thought that we could do a little bit better than this. So the rationale behind this is that rather than using the canonical gene or protein composition of a cell, where we see, you know, hundreds of thousands of genes or proteins that describe a cell state, we could move a step forward and look into cellular behavior. So really, here, these behaviors are, of course, conditioned by both the environment and the genes and proteins that are in that specific cell. We don't know in which way, but we know that a cell that is behaving in a specific way is integrating the presence of proteins, the presence of genes, and the influence of the environment. So can we read that information at the level of cellular behaviors to understand better biological output? So this is a question that we've been working for a while now. And the rationale behind this is now when we look at images, instead of just going for the parameter that we're interested in, imagine like if you're doing gene analysis and you're looking for the transcript of only one gene. Now you would say, why don't you do 10 genes? So why don't you do single cell transcriptomes and get hundreds of genes? So it's the same rationale. Why, instead of looking at only one parameter that gives us this information, which is really not very informative, why don't we look at 6, or we look at 15, or we look at 25 or 31, which is the maximum that we're obtaining in a specific experiment? You see how the distribution of the cell shapes very much as you gather information just by looking at the blunt cellular behavior of the cell, which is the shape of the cell and the movement of the cell. And we come up with this sort of information. We can, in this particular experiment, come out with a picture of the identity of the cell. So by looking at behaviors, we figure out the identity in this simple experiment where we could, we knew which cells were which, but we only look at that information afterwards. And we can discriminate the identity of, in this case, dendritic cells versus neutrophils. So the idea is now, can we use this in a more complex system? The complex system, again, is going back to our my old experiments with Paul in like 15 years ago, and repeat these experiments, take the parameters of movement and shape, analyze everything in an unbiased way, and come up with this sort of map, which is a heat map, which is a little bit maddening if you think about it, but we can start seeing already patterns here, patterns that differentiate this group and this other more general and abundant group and this other group, which have specific features that we can read. And of course, we can build, just like with single cell sequencing, we can build UMAP or TISNE maps, two-dimensional maps that explain the behavior of the cells, or explain the biology of the cells, or the states or the identity of the cells based on their behaviors. The great thing about this is that now we can apply this to acute responses in vivo, things that where we typically cannot extract the cells to look for the proteome or the transcriptome, but we can gather this sort of biological information. In this particular case, looking at the cremaster muscle when it's inflamed, and looking specifically at neutrophils, we see these transition states between these three different types of behaviors, for which we can define specific parameters, just like if you were looking at genes. We can look at how the length of the cell changes over time, or the volume at specific time, or whether the cell is in a position or another position if the velocity changes. So with all this, what we're doing is try to combine this with genetic models and try to identify the specific behaviors that will inform us about an important biological outcome. In our case, for example, acute inflammation, can we identify the key parameter that we should be looking at to identify ways to target that particular biological problem, for example, myocardial infarction. So I've discussed a bit of problems of heterogeneity and identity of the cells everywhere. I haven't entered into the bone marrow. There's a lot of work done here. I discussed mostly work in the blood, and I just wanted to go quite quickly, for the sake of time, into tissue. So what happens when the cell leaves the bloodstream and goes into tissues? And a lot of things happens. We generated a tool that we needed to generate in order to be able to track the cells once they leave the circulation. This is an inducible reporting under the OYCC locus, or neutrophil specific. So the system works. And with this tool and using a mathematical modeling, what we were able to estimate now is how long neutrophils live in the tissue. So this was an open question in the field, under the assumption that the cells die quite immediately after entering the tissue, they're eliminated or phagocytosed. There's nothing more to be interested in that particular cell type. But we thought, it was important to see really if the cells were eliminated immediately, and this was not the case. Of course, the cells don't live for days, but in steady conditions, they live anywhere from 8 hours to almost 20 hours in the case of the skin. So really, there's a lot of time for these cells to influence the physiology of the tissue. So again, for the sake of time, I will just go quickly. This was published anyway. The idea here is that we use an array of single cell bulk omic analysis to look at whether the identity of the cells in different tissues change also, very much like what's been described from macrophages. So we use site-of analysis, which I'm not showing here. We use single cell analysis, which already identify specific clusters associated with bone marrow and spleen, or lung or blood, and specific genes that were dominant in a specific tissue. So granule proteins that are synthesized in the bone marrow and spleen as expected, more inflammatory or immune tolerant related genes that are specific for the blood or for the lungs. So we also use bulk sequencing, which allows us to gain more depth and describe better the system. Again, we see diversity that confirms quite well with the single cell sequencing. Here, simply by projecting the signature in the bulk into the single cell, we see that there's a good match. So the two results are consistent, not an artifact of the system. We combine this with ATAC sequencing analysis that we run together with Irina Odalova at the UK. Again, to show that these changes in the transcriptional profile had an imprint in the chromatin of the cell. So even neutrophils actually live, they still have time to rearrange these. And of course, and again, doing a matching, checking that the transcriptome signature match well with this chromatin accessibility signature. So everything seemed to be in place and consistent. So these ideas allow us to play a little bit with the system, doing silico analysis like this one, taking data from Mikhail Pitet's lab, where he did single cell sequencing of neutrophils from the lungs of healthy or tumor-bearing mice, where he discriminated these two clusters. The question is whether the neutrophil, the lung signature of neutrophils is dominant or not over disease. A question that might be relevant for many different types of the inflammatory disorders. So what we found is that really the tissue signature, the lung signature, cannot discriminate between cells that come from a healthy or a tumor-bearing lung, indicating that what we think is the tissue signature is quite dominant, and it gives a strong imprint into the phenotype of the cell. And this is checking, as expected, no signature from the other tissues. So just to finalize a little bit, what we did as a summary of this is we ran these profiles now that we have confirmed that they were quite trustworthy, with a pathway analysis to predict what sort of functions could be predominant in its different tissues. So there are a couple of things that I will highlight here. One is that when you look at chemotaxis, migration, and immune response, which are the canonical functions for neutrophils, these are dominant mostly in the bone marrow and in the blood, which are the tissues which we have been looking at for decades. So in a way, it's not surprising that what we know about neutrophils is dominated by these migratory and immune functions, because we've been looking at tissues where this is the dominant function, and I guess for a reason. But if we start looking at other tissues, we start seeing pathways of functions that are particularly interesting and new, such as angiogenesis in the lung and intestine, or epithelial support in the skin. This is just a validation of this. Again, I won't go into details. This is just to show that if you eliminate neutrophils, then the recovery of leukocyte homeostasis is lost, as we predicted in our pathways analysis. And again, this is a new disguise for the neutrophil that becomes supporter of B cells, like a butler of B cells, or a supporter of stem cells in the bone marrow. So it allows recovery of the cells. If we look at the lungs, where we predicted angiogenesis, we see evidence that this really is the case, that they support the recovery of vessels, either if you give, not in the steady state we didn't see a difference, but if you now introduce a challenge with subrachal irradiation, or if you look at very young animals where this is still active proliferation, then the neutropenia creates a deficit in angiogenesis in both the lung and the intestine, but not in other organs where we did not predict this. So this is just a visual example of this. So this is the intestine, this intestinal isolated lymphoid follicles, which is where neutrophils accumulate, and where Ivan Ballesteros, who was leading this work, could see that there was an effect on vasculogenesis around these specific structures in the intestinal mucosa. And this is quantified here. So the effect is only seen in the follicles, but not in the rest of the tissue. So here we propose, we also identify neural support. So that's, we propose that the disguise of the neutrophil here would be for an electrician or a plumber. And finally, the hint, I think this is just thinking forward. One striking finding that we made also in this study is that when we focus in the lung, which was our favorite tissue because there are a lot of cells there, we could do a lot of analysis. It was easy, and so visually was quite easy to obtain good data. One thing that Ivan realized is that the distribution of neutrophils in this organ at steady state is not random, but rather associated to these CXCL12 positive expressing vessels. And this is the determination that this distribution is not random, and this a close association. So this raises the idea of a niche in the lung. So this is something that we're exploring, and I just wanted to bring up this idea because I think it's quite stimulating, even for a short-lived cell, that the idea that they might be niches or educating spaces. And this is consistent with an effect of the CXCR4 mutant, which is able to engage into this transcriptional profile obtained normally by neutrophils. So in the mutants, the cells don't adopt this profile. So that's what we think here, is that both CXCL12 and CXCR4, which is a ligand receptor pair, are needed to guide the cells right into the place they need to go in the lungs. So this is the neutrophil in disguise. This is the idea that it's a way to put the same idea of heterogeneity of functions, of fates, states, functions, as you want to put it, so that the cell is able to accomplish many more tasks than we thought they were able to do. So it's not only this antimicrobial effect in the blood that we've been studying for many years. Of course, it's essential, but many other functions tailored to the tissues. And this is the disguise, or this is the adaptability of the cell that is, we think, able of many more things that we thought they were able to do. In more of a conceptual note, the idea is that we see for decades we've been studying this unidimensional property of neutrophils produced from the bone marrow to fight infections and cause inflammation. More recently, we've realized that there's also functions of repair and tolerance, which become quite clear in the context, for example, of a tumor or a transplantation. And we propose by studying and giving a new angle to how we see neutrophils, we propose that there's a transition in the phenotype and functions of the cells, while in the circulation they enter tissues, and one in the tissues they acquire new functions, which are encoded here by colors. And again, we don't know still what many of these substances are doing in the tissues. And we think, you know, seeing this as a whole, that these more tolerogenic, these more inflammatory functions, what happens with the cells in the tissues, whether there are transitions between them, will be important to understand really what these neutrophils are and what the true disguise is. And just to make the note that this is granular and this is intentional, because this, as I showed before, there's a lot of state probably coexisting at a given time in the tissue. So this is the lab. Thank you to all of them. The work is mostly that of Jose Maria Adrover, Ivan Ballesteros, Alejandra Aroca, Georgiana Kronichuk, Miguel Palomino. So basically a coordinated effort from everyone. I'm very thankful to our funding sources, our fantastic collaborators. And before I finish, and I'm sorry for the extra time, I just wanted to announce that the lab will be moving to Yale. So Matteo mentioned this, and we will be recruiting for next year. So please contact me if you're interested in, you know, helping out our good friend, the neutrophil, come out of this identity crisis and many other fascinating questions in physiology. So thanks so much for listening.
Thank you, Andres. This was terrific. Thank you. Very insightful research on neutrophil biology, from the circadian regulation, heterogeneity, and all the many tasks that neutrophils play a role in. Yeah, thank you so much. It was very illuminating. Thank you.
Thanks so much. Thanks so much.
And congratulations on your move to Yale.
Thanks very much.
Best of luck. Okay, so I will share my screen now. So I can guide all the attendees to ask questions via Twitter. And so for that, you will have to go and search for the account Global IMMUNOTALKS and find the tweet that says ask questions for Dr. Andres Hidalgo here, and reply to that tweet with a hashtag Global Immuno, and then Andres will be using his personal Twitter account to answer questions. And remember, the reason why we do questions via Twitter is so they can be asked and answered in an asynchronous manner. And so if you have questions when you watch the talks in YouTube, you can also post them in Twitter. Okay, so with that, we will close the session, and we hope you join us again next week for the Global IMMUNOTALK by Matteo. Thank you again, Andres, and hopefully see you soon.
Yeah, I count on it.
Take care. Bye, everyone. Thank you.